Hi guys, this video is our mid-semester 2 revision video, and I'm gonna try to make it as much as I can in the form of questions and answers. Concerning the first slide, which is slide number 1, of course, the diagram to the left that represents the neuron is very important. So I might get the diagram and ask you, what does the diagram represent? So the diagram represents a typical neuron. Of course, you can add that it represents a typical myelinated neuron because, of course, you can see that the axon is covered by a myelin sheath. Now, when we come to the structure of the neuron, the detailed structure of the neuron, you might be asked several questions. You might be asked about the mature nerve cells. Are the mature nerve cells active cells? Do they actively divide? No, they are resting cells. So the mature nerve cells do not divide. They do not enter in cell division in order to keep their function and to keep the neuronal connections intact and unchanged. Coming to the dendrites, I might ask you to define the dendrites. So the dendrites are short, highly branched, multiple processes that are connected to the cell body of the nerve cell. And the function, what is the function of the dendrites? The dendrites receive information or receive impulses from other neurons. So they always receive. And you cannot say that they receive sensory impulses or motor impulses, because this neuron, that's most likely a motor neuron, might receive an impulse from a presynaptic motor neuron, because there is a synapse in between them. So you just say that the dendrite receives impulses. And of course, the cell body contains the nucleus. The nucleus in nerve cells is highly developed, because when I said that the mature nerve cell is not an active cell, I don't mean that it's not active in metabolism and in metabolic processes, but I mean that it does not actively divide. So we would rather make it more clear. The cell body of the neuron does not actively divide. The nucleus does not actively divide. But on the other hand, the neuron, of course, most neurons are highly active. The biochemical processes inside the neurons are very high, because all these electric impulses that are generated at a very rapid rate, of course, necessitate a very active rate of metabolic processes inside the cell. Now we come to the other process or the other branch, because as you know, the typical neuron has short branches on one side, which are the dendrites, and a long process on the other side, which is the axon. And the axon is terminated by branches that are called terminal arborizations. And arborizations comes from the radical of the word is like arbre or arbre in French, which is a tree. So it's like the branches of a tree. So we call this in general is called axon terminal, but the branches themselves are terminal because they are present at the end, arborizations. You might also be asked to compare and contrast between the axon and the dendrites. So the dendrites are short, the axon is long. The dendrites are multiple, the axon is single. We're not talking about the terminal branches, we're talking about the axon itself. The dendrites are receiving all the time impulses, whereas the axon is sending impulses peripherally or away from the cell body. Of course, I might ask you if the nerve transmission in the axon is fast in this axon in particular, in that diagram in particular, is it fast or slow? Of course, when I ask such a question, it's an indirect way of asking you about the myelin sheath. Of course, since you see the Schwann cell and you see the myelinated segments, then you will say that the conduction in this axon in particular is very rapid. And you have to justify why it is rapid, because the axon has myelinated segments. And when there are myelinated segments, this means that in between there are unmyelinated segments that are called nodes of Ranvier. So what are the nodes of Ranvier? They are unmyelinated segments on the axon that lie in between the myelinated segments or the segments of the myelin sheath. And the presence of this structure of the myelin sheath is a good adaptation for rapid conduction, the rapid type of conduction, which we call saltatory conduction. And it is, how does saltatory conduction occur? The electric impulses jump from one node of Ranvier to another. This is because the myelin sheath is an insulator sheath. It's made of fat, and the fatty material is insulator. And since it is insulating, then the electric impulses cannot pass through, and they will jump from one node of Ranvier to the other. I might also ask you a more complicated question, saying, for example, how can you explain the paradox between having the myelin sheath being made of fat and being an insulator or a poor conductor of electricity, and at the same time that myelinated axons have a rapid rate of conduction? In such a case, you would say that this insulation helped the electrons to bypass the insulating region and jump from one node to the other. If the myelin sheath were not there, there wouldn't be the saltatory conduction. It would be the regular conduction of electric impulses along the axon, which is much slower. I might also ask you a more difficult question about the Schwann cell and the role of the Schwann cell around the axon. Of course, first of all, the Schwann cell is responsible for formation of the myelin sheath in the peripheral nervous system. And it is equivalent to the oligodendrocyte that makes the myelin sheath in the central nervous system. How does the Schwann cell form the myelin sheath? By stretching out itself to the maximum and rolling around the axon like a Swiss roll, and making concentric layers of its cell membrane. Because of course, when it turns like a Swiss roll, there would be several layers of stretched out cell membrane around the axon, and every cell membrane is a phospholipid bilayer, so it mainly consists of lipid molecules, and that's why the myelin sheath is an insulator and it's made of fat. But the Schwann cell has another important function other than just making the myelin sheath. The Schwann cell helps in repair of the axon. If the axon is cut because of a mechanical injury, for example, then the Schwann cell will de-differentiate, DE, and then differentiate. De-differentiate means that it will return undifferentiated, and it will start dividing to form a sheet of cells that joins the two cut end of the axon, but of course, provided that the two ends of the axons are close enough to each other. I think the last question I might ask you about the diagram would be, for example, where can you find the myelin sheath, or where can you find such neurons? Of course, you can find such neurons whenever you have white matter. Why am I saying this? Because the axon is myelinated, and the myelin, being fat, gives a shiny white appearance. Of course, I'm not talking about the stained myelin sheath under the microscope. I'm talking about the natural color of the myelin sheath. It gives it a shiny white appearance. So you would find such axons such myelinated axons in the white matter of the cerebral hemispheres, in the white matter of the spinal cord, around the central H-shaped gray matter. You would also find it in motor nerves, peripheral motor nerves, because of course you know that the motor nerves are nothing but a parallel collection or groupment of motor neurons. Of course, there is a great difference between a neuron and a nerve. Here we are talking about neuron. We are talking about a neuron, meaning that we are talking about an individual nerve cell and not about a nerve, which is a collection of parallel or neurons arranged in a parallel manner. I might also ask you about these swellings at the end of the terminal arborizations, and these swellings are called the synaptic knobs, and about their importance. Of course, the synaptic knobs are swellings present at the ends of the terminal arborizations. They're full of synaptic vesicles. These vesicles are full of neurotransmitters, and the neurotransmitters are what? The neurotransmitters are chemical substance, neurohormones, that are present in the vesicles and that are released when these vesicles approach the cell membrane of the synaptic knob and rupture. But in order for these vesicles to approach and rupture and release the neurotransmitter in that space, that is called the synaptic cleft or the synapse simply, the electric impulse should arrive. So upon arrival of the electric impulse, calcium is pumped into the synaptic vesicles. The synaptic vesicles move towards the membrane of the peripheral membrane of the synaptic knob. The neurotransmitter, they rupture, of course, the neurotransmitter is released and the neurotransmitter will diffuse across the synapse. Waiting for the neurotransmitter on the other side, there is one of three structures. And that's why here we can have questions about, but I'm postponing these questions until we reach the synapse. But for the moment being, you must remember that a neuron might synapse with another neuron, or in a synapse, of course, or it might synapse with a gland, or it might synapse with a muscle, whether it is a skeletal muscle or a smooth muscle, and we call all the muscles and the glands, we call them effector organs. I might also ask you about the nervous system components, but I might call them nervous system organs instead of nervous system components. So don't be amazed if you find the word organ, because the brain is an organ, the spinal cord is an organ, and the nerves themselves are organs. So first of all, if I ask you about the nervous system components, I mean here, in that slide in particular, I mean the nervous system divisions. So we have the central part of the nervous system, which is the brain and the spinal cord, and we have the peripheral nervous system. The peripheral nervous system is all the nervous structures that are outside the brain and the spinal cord. So you have 12 pairs of cranial nerves. So that arise from the brain, of course. So the total number of the cranial nerves, if I ask you about the total number, because you have complete questions in the test. So sometimes the complete questions, the blanks, are more difficult to answer than the essay questions or the free response questions. So the total number of cranial nerves is 24 because you have 12 pairs. The total number of spinal nerves is 62 because you have 31 pairs of spinal nerves. If we were to compare from that slide only, from the data on that slide only between the cranial nerves and the spinal nerves, we would say that we would talk about the origin first. The cranial nerves arise from the brain. The spinal nerves arise from the spinal cord. This is the origin. The number of cranial nerves is 12 pairs, and the number of spinal nerves is 31 pairs. And then we might also add something that's very useful about the nature of those nerves. The cranial nerves can be either purely sensory, like the optic nerve, like the olfactory nerve, like the auditory nerve, or they might be purely motor, or they might be mixed, like, for example, the facial nerve. But we have all the spinal nerves are mixed, and we will see why. Because when we studied the reflex arc, we learned that the spinal nerve, that is a mixed nerve, has two roots that connected to the spinal cord. And that was the question on the chapter test. It has a dorsal root that is sensory and a ventral root that is motor. But these roots, as we will see on the diagram later, they separate only close to the spinal cord, but away from the spinal cord in the periphery, they are together in the same nerve membrane or enclosed by the same nerve sheath and nerve membrane. So the spinal nerves are all mixed. And when we say mixed, we mean that they have both sensory and motor fibers. Here you have the diagram of the central nervous system, the brain and the spinal cord, and you have the peripheral nerves that arise from the spinal cord. Some of them are very long, like the sciatic nerve, for example. But in the brain, why didn't we draw the cranial nerves? Because most of the cranial nerves are very short, because they arise in structures that are very close to the brain, like the ear, like the nose, like the eye, and they reach centers in the brain. They are mostly not seen in a diagram like this, but of course, they are seen in real anatomy when you open the brain and see the connected nerves. In the peripheral nervous system, we have ganglia. Just like in the central nervous system, we have nuclei. So the ganglion is a collection of nerve cells of nerve cell bodies, not nerve cells in general. The ganglion is a collection of nerve cell bodies that is present in the peripheral nervous system. Whereas the nucleus is the equivalent of the ganglion in the central nervous system. The nucleus is again a collection of cell bodies, of nerve cell bodies. But instead of saying in the peripheral nervous system, we will say in the central nervous system. And we have seen some of these nuclei, like the superior olive, like the colliculus, and other nuclei, the medial geniculate nucleus that we studied in the pathways. Okay. Now we come to the lateral surface of first of all, the components of the brain. I might also ask you about the organs that make up the nervous system. So you would say that the central nervous system is made of two main organs, the brain and the spinal cord. And you would say that the peripheral nervous system is made of cranial nerves and peripheral nerves, because the nerve is considered an organ because it's made up of several layers of tissues. For example, as we will be discussing later on, there is the nerve sheath or the neurolemma around the nerve. There is the connective tissue inside the nerve in between the axons. There are blood vessels. Just we were describing a muscle bundle, for example. The nerve is considered an organ. Now, the human brain consists of three parts, as you know. So what are the parts that form the human brain? This might be a short answer question, or it might be a complete question. So it consists of the cerebrum, which itself consists of two cerebral hemispheres. It consists of the cerebellum, and it consists of the brainstem. And the brainstem consists of the midbrain, the pons, and the medulla. And then we have the cerebellum at the very posterior part of the human brain. Now, in every cerebral hemisphere, you have to know the relative positions of the lobes, because you might be asked to label the diagram, or you might be even asked to draw the lobes on the cerebral hemisphere. So first of all, if you are asked to draw the cerebral hemisphere, then you will start by making this fissure, which is the lateral sulcus or lateral fissure, and then you draw another horizontal fissure that separates the frontal lobe from the temporal lobe. You needn't know its name now. So this lateral sulcus or fissure separates the frontal lobe anteriorly from the parietal lobe posteriorly. If I ask you about the function of this sulcus, whereas this transverse sulcus between the frontal lobe and the temporal lobe, or this horizontal sulcus, separates the frontal lobe from the temporal lobe anteriorly and separates the parietal lobe from the temporal lobe posteriorly. And then you have to draw another sulcus posteriorly that separates the parietal lobe from the occipital lobe. So you start by drawing the three sulci. And then you write frontal lobe, parietal lobe, occipital lobe, temporal lobe, and then you draw the motor strip and the sensory strip. Why are we drawing it if we are not asked essentially to draw the centers in the drawing? We're just drawing them in order to be able to localize or determine the location of Wernicke's area and Broca's area. So you will draw Broca's area, the middle part of the frontal lobe, just anterior to the motor strip, and you will draw Wernicke's area in the posterior part of the superior temporal gyrus, because you know that a sulcus is a fissure that divides the lobe into gyri, and that the gyrus is the elevation between two sulci. So if you have a superior temporal sulcus here, and then another temporal sulcus here, so in between them, we will have three gyri: superior temporal gyrus, the middle temporal gyrus, and the inferior temporal gyrus. And the Wernicke's area is present in the posterior most part of the superior temporal gyrus. You might also be asked about the anterior most part of the cerebral hemisphere. It is the frontal lobe. This is called the frontal pole. So this is the anterior most part of the cerebral hemispheres, and the posterior most part of the cerebral hemisphere is the occipital lobe. And then here you have the cerebellum just underneath the occipital lobe and the temporal lobe. This represents the inferior surface of the frontal lobe and of the cerebral hemisphere. And I might ask you about this inferior surface, for example, to point an arrow at the inferior surface of the frontal lobe, and at the same time to tell me what cranial nerve passes underneath this inferior surface of the frontal lobe, or related to this inferior surface, and it would be, of course, the olfactory nerve, as you know, the olfactory tract, actually, not the olfactory nerve. This is the superior surface of the cerebral hemisphere. Now you also have to know the functions or the centers that are present in every lobe. So of course, you know that in the occipital lobe, we have the vision center that is responsible for receiving electric impulses from the optic nerve, and the vision association area that is responsible for understanding what we see, which we call perception of vision. In the temporal lobe, and in particular in the superior temporal gyrus, and most anteriorly, we have the auditory center, and behind it, we have the auditory association center. The auditory center is just responsible for detecting or receiving sound stimuli. When I say detecting, this is not understanding. Detecting means that you just realize that there is a sound, but behind it is the auditory association area that's responsible for understanding those sounds. You hear a particular sound, you see, you say, you understand that this is your mom's voice, this is your dad's voice, this is your teacher's voice, this is a particular singer's voice, and so on. And then we have, of course, the Broca's area and the Wernicke's area, and we've talked a lot about Broca's area and Wernicke's area. I'm not sure whether they are on the slides or not, but we wrote them in the notes. So Broca's area and Wernicke's area are two important centers for formulation of speech. And when I say formulation of speech, I mean formulation of the ideas of speech, like Vernix area. Understanding the meanings of words and the meaning of the speech that we are hearing or listening to. So Vernix is essentially sensory, but Broca's is essentially for articulating words. So when you say formulating words, you mean formulating ideas of words. But when you say you want to say pronouncing words, you would say articulation of words. Damage to the Broca's area or damage to the Vernix area will result in disturbance of language, disturbance of spoken and written language in case of a damage to Broca's area, or a disturbance in understanding the meanings of words. Like, for example, when you're reading a story in the classroom, or you're reading a comprehension passage, or listening to your teacher, or listening to your parents, you wouldn't understand what they say. So aphasia is a disturbance in speech and or a disturbance in language. Broca's aphasia is called motor aphasia, and Vernix aphasia is considered sensory aphasia. Broca's aphasia why is Broca's aphasia considered motor aphasia? Because there is lack of coordination between the muscles of articulation. The muscles of articulation include the muscles of the lips, the muscles around the lips, the muscles of the tongue, the muscles around the larynx. All these are muscles of producing sound, speaking, and articulation at the same time. Broca's area thus is responsible essentially, if you don't have aphasia, then Broca's area would be responsible for coordination between the voluntary muscles of articulation and coordination. This is for articulation, but for writing, you would have coordination between the fine muscles of the hand, the fine movements of the small muscles of the hand. Broca's area is also responsible for proper or correct structure, grammatical structure of the sentence. And therefore, in case of Broca's aphasia, the grammar would be disrupted. The patient does not use prepositions, does not use pronouns, does not use linking words. And that's why this is another justify question. The first justify question, or the first justification question, was why is it motor? But the second one, why is Broca's aphasia considered non-fluent aphasia? It is considered non-fluent aphasia because, as you know, the speech is telegraphic speech, and the patient gives interrupted, non-fluent, non-smooth syllables. And the patient is very frustrated, of course. Justify why is Broca's, or why are Broca's aphasia patients usually frustrated, because they understand that they have a problem in articulation and that they cannot say what they want to say. Vernix area is considered sensory because it's not at all related to the muscles of articulation. It is related to understanding the meanings of words, formulating the ideas of speech. Of course, if you don't understand the words, you wouldn't be able to use words properly in a sentence. But on the other hand, because Vernix is essentially sensory, is essentially a problem. And when I say sensory here, I mean a problem in understanding. So the patient is not aware that he has a problem. So they're not usually frustrated, and they're not careful when they talk, because Broca's patients are careful when they talk. So they're not fluent, but they're not fluent because they are aware of the problem and because they do not coordinate. That's why they're not fluent. In Vernix, first of all, they are not aware that they have a problem. And second, they do not have a problem with coordination. Their coordination is okay, so they give you fluent speech, but it is nonsense speech, and they invent new words. Concerning the higher functions of the frontal lobe, they are mainly very sophisticated. They are related to the most difficult functions that a human brain can do, which are the reasoning skills, the decision-making skills, the logical skills, and behavior, especially in the anterior-most part. Of course, when you know the function of an area, you will be capable to know what results from damage of that area. In the frontal lobe, we don't only have, that's why we said in the very anterior part. So in the very anterior part of the frontal lobe, you have centers that are responsible for decision-making, for logical skills, and for behavior. And for habits, the memory of habits is also present in the frontal lobe. They are not written here, but they are written later on. The memory of habits, motor habits, like, for example, driving the car, like making coffee, and so on. So if there is damage to the anterior part of the frontal lobe, then you will lose all these skills. Definitely, if the question is a 3D question, I might ask you, what are the causes of Broca's aphasia and Wernicke's aphasia? What are the causes of frontal lobe lesions? So in this case, you should use all the possible knowledge that you know about the lesions that might affect the brain. So for example, it might be a benign tumor, it might be a malignant tumor, it might be a stroke, a vascular accident. A vascular accident might include hemorrhage, or it might include a thrombus. But the frontal lobe, as we said before, in the very posterior part of the frontal lobe, because here you have this lateral sulcus, the superior part of the lateral sulcus separates the motor strip anteriorly from the sensory strip anteriorly. So part of the frontal lobe is responsible for giving motor orders to the opposite side of the body. So if this motor strip is damaged by any of the reasons by any of the causes we have mentioned before, then there will be paralysis of the muscles on the opposite side of the body. But of course, the extent of the paralysis will depend on the extent of the motor strip. If the whole of the motor strip injury, if the whole motor strip injury, the whole motor area is injured, then the paralysis will involve the whole half of the body. In the parietal lobe, in this slide, we're talking only about the sensory area. Of course, you have a sensory area, and besides it, we have a sensory association area. The sensory area is responsible for receiving all the ascending sensory stimuli that come from the lower parts of the body, either from the spinal cord or the cerebellum, or even from the brainstem. And of course, you know that here, but we don't see it because it is in an internal structure. You have the thalamus. And we said that the thalamus is the secretary of the brain. We're going to study the thalamus in detail. But we have said only one important thing about the thalamus, which is the secretary of the brain, that all sensations, all ascending sensations from the lower, from the parts of the body lower than the brain will be relayed in the thalamus. They should pass through the thalamus to be filtered, because sometimes the thalamus finds that this sensation is not important. There is no need to give it to the brain. It does not need a higher control or a higher decision. But in the parietal lobe, we have the sensory association area, and the sensory association area of the parietal lobe is not only about understanding all these sensations that are received by the sensory area, like, for example, the pain sensation, temperature sensation, even proprioception and position of the joints, but the sensory association area here helps you first perceive all these sensations. Perceive is the verb from perception, but it's also important for integration of sensations, because, for example, we will study later on that the parietal, or we will see later on, we already studied this on other slides, that the parietal lobe, for example, is important for math skills. It is important for sequencing and arrangement of things, for recognizing objects and faces, and so on. So there is a kind of integration, because when you touch something, for example, and you touch something, so you're using your touch sensation, which reaches the sensory strip in the parietal lobe, but you also have to recall what this represents. So the parietal lobe is associated, for example, or connected to the hippocampus that is responsible, as we saw in the comprehension, for recalling sensation. What I want to say is that the sensory association in the parietal lobe is far more complex than just understanding the classical sensations that reach the sensory strip. The cerebellum, of course, is a very important organ, because the cerebellum is important for two major functions: coordination of muscle movements, meaning, for example, if an agonist is contracting, then the antagonist should be relaxing, and vice versa. And because of this coordination, that is specifically important in fine muscular movement. Where you need the most the coordination of the cerebellum is when you have fine, small muscles that have to contract and relax, and at a very rapid rate, like, for example, the small muscles of the fingers, the muscles of the face sometimes, but the typical example is typewriting or playing the piano. So if you have a cerebellar disease, you wouldn't be able to coordinate these fine muscle movements. If you are a piano player, you would be no more able to play the piano. You would be no more able to typewrite or to use the keys on the keyboard of the laptop. You wouldn't be able to do fine coordinative movements like buttoning your shirt or unbuttoning it, and etc. But it is also important for balance and posture, because we saw that there is a connection between the semicircular canals in the internal ear and the cerebellum. And the connection is the 8th cranial nerve. So if I ever ask you about how the cerebellum maintains balance and posture, or I might ask you a more difficult question, what's the connection between the ear and the cerebellum? The connection between the ear and the cerebellum is actually a connection between the inner ear and the cerebellum. But the inner ear contains two organs, the cochlea and the semicircular canals. So it's actually between the semicircular canals in the inner ear and the cerebellum. The nerve that connects the ear to the cerebellum is the 8th cranial nerve. But the 8th cranial nerve has two divisions. What are the two divisions of the auditory of the 8th cranial nerve? Actually, they are the auditory division and the vestibular division. Or instead of saying auditory, we would rather say cochlear division. So the nerve is called vestibulocochlear nerve, the 8th cranial nerve. So justify, why is the 8th cranial nerve called vestibulocochlear nerve? Because it has two divisions. A cochlear division for hearing, coming from the cochlea, the organ of hearing that we saw in the document I've just sent to you on Google Classroom today, and we discussed it on Thursday, where we had the drawings for the cranial nerve pathways, and there is the semicircular canals that are connected via the vestibular branch of the 8th cranial nerve. What happens is that when you are about to lose weight, you lean forward, and the semicircular canals have liquid inside, so the liquid moves, and inside we have the sensory receptors, which are nothing but the nerve endings of the cochlear division of the auditory nerve, or the 8th cranial nerve. These hair cells will be stimulated, and as you know, any sensory receptor will convert any type of energy into electric energy. The movement of the liquid inside the semicircular canal, this is what? This is kinetic energy or mechanical energy, and it will be converted into electric energy transmitted via the auditory nerve, of course, the cochlear division or the auditory division of the 8th cranial nerve, to the cerebellum. The cerebellum realizes that the position of the body has changed, so the cerebellum gives orders automatically via descending cerebellospinal pathways. There are fibers that descend from the cerebellum to the spinal cord. This is the higher pathway, and then from the spinal cord horizontally along the motor nerve to the muscles of the back to contract. Of course, this is a reflex. We have the receptors in the semicircular canal. We have the auditory nerve, which is the afferent nerve that carries the sensation to the cerebellum. We have the center of the reflex, which is the cerebellum. And then we have efferent descending motor nerves from the cerebellum to the spinal cord. And then we have the lower motor neuron that supplies the anti-gravity muscles, giving order to those anti-gravity muscles to contract, and you raise your back again, you stand up erect again, and the problem is solved. And this is a major example to show you how reflex actions, which are automatic, involuntary actions, first of all, can have centers other than the spinal cord. Not all reflexes have their centers in the spinal cord. Here we have a center in the cerebellum. The center of the reflex is in the cerebellum. And at the same time, it shows you how these automatic involuntary responses are protective to the body, because we have always taught our students while explaining reflex actions that they are protective to the body. But here you've noticed that the brain wasn't involved. The cerebral hemispheres were not involved. But this doesn't mean that the cerebellum will not send impulses to the brain. Maybe the brain would take more actions. But first of all, the cerebellum takes a very rapid action in order to save the situation. And then the brain afterward can take further, more complex actions. Concerning the brain stem, the part that is shown just under the surface of the cerebral hemispheres and just above the spinal cord is the medulla oblongata or medulla oblongata. Above it, we have the pons, and then we have the midbrain. Usually, you can't see the midbrain in a section like this. It's very difficult unless you elevate the temporal lobe to show the midbrain, and then the pons, and then the medulla. And notice that the medulla is continuous with the spinal cord. Of course, you have to know the order. You have to know, because you might get a diagram like this and label. So you have to know that the midbrain is the uppermost, the pons are in the middle, and the medulla is lowermost. Of course, the brainstem is very important, because if you look at that diagram, you would see that any fibers that arise from the cerebellum and want to go to higher centers in the brain will pass through the brainstem. And on the other hand, any descending fibers from the brain to the cerebellum would again pass through the brainstem. That's why we say that the brainstem acts as a relay center that connects the cerebrum to the cerebellum, and also connects, if we imagine that the cerebellum is here, then it will connect the cerebellum to the spinal cord. The brainstem, of course, controls especially the medulla oblongata, because most of these, the nuclei or the collections of nerve cells, usually when we say nuclei and when we talk about nuclei inside the central nervous system, we talk about a collection of nerve cells, of nerve cell bodies in particular. Just like the ganglion is a collection of nerve cell bodies in the peripheral nervous system, the nucleus is a collection of nerve cell bodies in the central nervous system. And usually we talk about, why is it important to talk about the cell bodies? Because the cell bodies are considered the brain of the neuron. They are the part that integrates information, that makes decision for a single neuron. So when you have a collection of cell bodies that are close to each other, connecting to each other, this is a very high level of synapsing first and of integration. So the nuclei are very important for synapting and for integrating. Most of the nuclei responsible for these automatic involuntary actions like involuntary breathing. I'm not talking about controlled breathing. I'm talking about the subconscious breathing that we do all the time without being conscious to it. The heart rate, digestion, sneezing, coughing, vomiting, and swallowing, all these are reflexes and mostly present in the medulla oblongata. That's why whenever the medulla oblongata is injured, whether accidentally by a traumatic accident, for example, a vertebra, a broken vertebra that sharply cuts the medulla during an accident, or voluntarily or through a hit, for example, on the back of the neck, then the patient would die. This is lethal. Why is it fatal and lethal? Because you are damaging the centers of breathing. You're damaging the centers that control the heart rate. And of course, if the heart does not beat and if the person does not breathe, then they would definitely die. When we talked about the main lobes of the brain, we talked about the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And we said that there is a fifth lobe that's embedded in between the four lobes, and that you can see only when you retract the frontal lobe and you retract the temporal lobe, which is the insula. Of course, the insula has many functions, but the main function of the insula that it contains the gustatory center or the center of taste. That's why if you look here, you have the gustatory cortex, which is this purple cortex, and around the gustatory cortex, you have the gustatory association cortex or taste association area. And here you can see the olfactory cortex. When we were talking about the hippocampus and the olfactory area, which is the final destination of the olfactory nerve, because you know that the olfactory tract moves on the under surface of the frontal lobe and ends in the medial surface of the temporal lobe. And this is, now you can understand why we were saying on the medial surface. When we say on the medial surface, this means that you cannot see it from the external surface of the brain. You have to retract using a retractor and look underneath, look at the inner surface or the medial surface of the temporal lobe in order to see the olfactory area, okay? We will stop here and I'll be sending this part of the video and then I'll be sending another part very soon, inshallah.